Okay, sorry about that. I was just showing you that the sperm and the ova, the gametes, are haploid. So they contain half the amount of genetic material. And this is because it's very important that during reproduction, these two cells are going to fuse together to form the zygote. The zygote is the fertilized egg cell. Okay, so the fertilized egg is called a zygote, which is going to develop into a blastocyst and then an embryo and then a fetus. So it's very important that these cells are haploid so that when they fuse together, they have the full amount of DNA. They become diploid. So your zygote, your fertilized egg, is diploid. Okay, so it has a full set of chromosomes. So, and that's very important. So if meiosis went wrong and they didn't have half the DNA in each one, if it had a full set and it was diploid, then they may not be able to fuse, or it may lead to genetic conditions because there's too much DNA there. Instead of having a diploid zygote, you might have one that's got more than two sets of DNA. Okay, so that's what we're talking about with the different types of cells and some of the terminology for you to remember especially. So diploid is just the number, is having two sets of chromosomes in your cells, and all of our bodily cells contain two sets of chromosomes except for those cells specifically in the reproductive tissue which we just spoke about. Okay, so most of our cells are diploid. They have a double set of chromosomes, and we identify with different species according to their diploid number. Okay, so we will look at how many sets of chromosomes that they have on a diploid level, because each organism has a different diploid number. That makes sense. So we've said why this is important. One of the most important processes during meiosis is obviously the exchange of genetic material between the maternal and the paternal chromosomes. So the maternal from the mother, paternal from the father. They're homologous, they're similar but not identical. But when they come together, there will be exchange of genetic material, and this is what produces variation in the offspring. So this is why children often have some characteristics from their mother, some from the father. They might have some that are different to their mother and father as well, and that's because they're not expressed in their parents. So this is where the genetic information is really mixed up during meiosis. So we have the exchange of genetic material, and some of that genetic material may not be expressed in your mother or your father, so you may not see it, but it's there in their cells. So you might receive that as a child and be completely different in some ways. So, and some organisms will develop what we call adaptations, which enable them to be better suited to their environment. And over a longer period of time, this obviously leads to evolution, which we'll come on to later on. So this all comes from the process of meiosis. It's very important, this exchange of genetic information that takes place. We will go through both meiotic divisions, but it's important to remember, I think, that meiosis 2, or the second meiotic division, is very, very similar to mitosis. So if you've remembered mitosis, meiosis 2 is very similar. It just has half the amount of DNA. Half the number of chromosomes. Okay, so it makes it a little bit easier to remember. So we'll go through that one now. Been through all of the other diagrams, I believe. Okay, so it's quite long, but so meiosis 1, you need to remember 5 different parts. Okay, so again, apologies for my diagrams, but you might want to draw your own, because it's a good way of remembering them. It's a good way of revising, is if you just sketch it, even if you're bad at drawing like me, sketch something. It's going to help you remember and help you to revise. Okay, so number 1, as we said before, interphase. We consider interphase to be at the beginning, and this is where DNA replicates itself. So it's before meiosis can take place. The DNA again has to replicate. So within the germ cells of the testes and the ovaries, the DNA will replicate. Those are diploid cells, and they will replicate their DNA. Okay, as I put at the bottom, it's not part of meiosis yet. And then we have prophase 1. Okay, so during prophase 1, your nuclei and your nucleolus will disintegrate, the same as in mitosis. You also get your spindle fibres forming, same as in mitosis. Chromosomes will coil up and condense, the same as in mitosis, but this time we get synapsis occurring. So synapsis is pairing. It just means pairing up of homologous chromosomes. So homologous, remember, means similar but not the same. So homologous chromosomes will pair up with each other during prophase 1, and that's called synapsis. Okay, and tetrads will form. That means groups of 4. So you'll see here you've got 1, 2, 3, 4, 1, 2, 3, 4. You can see I've got 2 red ones and 2 blue ones, 2 red ones and 2 blue ones. So they arrange themselves in groups of 4 during prophase 1, and these are called tetrads. Tetra means 4. Okay, and when they're in these tetrads, that's when we start getting some of the genetic exchange of information taking place, so which we call crossing over. So the crossing over will start during prophase 1. Okay, the next one is metaphase 1, and this is where your tetrads will align themselves at the equator, again like in mitosis, but your tetrads are aligning themselves at the equator. And here we will get independent assortment, which is another type of exchanging genetic material other than crossing over, where you just get some material crossing over at the chiasmata. With independent assortment, this is where they randomly separate from each other. So chromosomes will randomly separate. That's what it means, independent assortment. And we also get some crossing over during this stage as well at the chiasmata. The chiasmata holds the 2 sister chromatids together and allows the crossing over to take place, and that happens at the chiasmata. It's important to remember that for your exchange of genetic material. So it's all happening during metaphase 1. Very important. Independent assortment and crossing over continuing. Then we get to anaphase 1, which again is similar to mitosis, but this is where your homologous chromosomes will actually separate and move towards the poles. Okay, so your homologous chromosomes separate and move, they're pulled by the spindle fibres towards each pole, just like in mitosis. Okay, so you've got the 1, 2, 3, 4 red ones up here, 1, 2, 3, 4 blue ones down here. Okay, and then we have telophase 1, which is where each pole now has a haploid set of chromosomes. So each pole is started to divide into 2, the cell. Each pole now has half the set of chromosomes, because in the beginning